Hi, everyone, and welcome to this British Science Week live lesson, CSI Seabed. We're going to be finding out how eDNA can help to support conservation, and we're going to focus on the amazing seagrass habitat. Before we start, I'm just going to give a few shout outs to some of those schools who are joining. Fantastic number of schools with us today. We've got a shout out to Level 1A from Uxbridge College, London. Hi, everybody there. Hello to the students in 5D, and they are at RGS Prep Guildford. To the amazing students at Homewood House School, and that's from everyone's favourite teacher, who says thank you for being so awesome. And last but by no means least, we have Mrs. Nicol and 8X1 from St. John Henry Newman Catholic School, and they are very much looking forward to learning how seagrass can help tackle climate change. Now, this live lesson and the wider Ocean and Climate Education Programme is part of the Convex Seascape Survey. That's a 5-year international science project, and they're looking at one of the least explored habitats in terms of our world, but also in terms of climate change, and that is the continental shelf. Now, you can perhaps see a map up on screen now, and you can see the pale blue bits coming out from land just before getting deeper into the ocean. That's the continental shelf, and it's an area which absolutely huge. Sort of, if you think in terms of countries, it's 3 times the total area of the USA and 4 times as big as well-known habitats such as the Amazon rainforest. And what we're really interested in is how much carbon is stored there, because storing carbon away from the atmosphere is a great way of tackling climate change. And to take us further into all these questions, eDNA, CSI Seabed, seagrass and everything, we're delighted to be joined by Richard and Bex from the University of Exeter. Hi, both of you. How are you?
Yeah, good. Thank you, Jamie.
Yeah,
Hi, everyone.
thanks.
Fantastic. You're both working as part of the Convex Seascape Survey. You've been doing some amazing work, which we'll get into a bit later. But we're just going to take us classes through these big questions sort of step by step. And the first thing is really just have a sort of an overview. I talked about how the seabed can tackle climate change. Can we just go through the carbon cycle basics a bit again, just so we get an understanding of the role of the seabed in this?
Yeah, sure, absolutely. So in terms of the land carbon cycle, really for us, it's sort of vegetation very much sits at the middle. So you've got your plants, your vegetation there, central to that carbon cycle on Earth. And you've got photosynthesis happening in the day when you've got sunlight, that process really of the plants are doing to grow. That's taking in CO2 or carbon dioxide, carbon from the atmosphere during the day. And then at night, those plants are respiring or breathing, releasing CO2 back into the atmosphere. So you have this cycle going on between the atmosphere and the plants. You also then have the soil. Obviously, plants grow in soil, and some of that carbon ends up in being sort of locked away and stored within the soil. But also you've got plants and animals and even us, that when we die and decay, that's the carbon that's held within those organisms goes back into the soil and ends up in those stores there. And then over geological time, so we're talking millions of years, some of that plant material actually then forms fossil fuel. So it decomposes further, goes under lots of pressure, and becomes these fossil fuel stores that we hear so much about.
We haven't been looking after that long-term carbon storage terribly well.
No, that's right. So humans have been sort of significantly using fossil fuels, so oil, coal, gas, for the past 250 so years. That's powering our homes, fueling our cars, and providing materials for sort of a large proportion of our everyday objects. So plastics, for example, are a product of fossil fuels or of crude oil. And while you can argue that fossil fuels is sort of fundamental to what we sort of envisage as sort of modern civilization, the rate at which we're using these fuels has been, and we're extracting them and burning them, is releasing that carbon far greater than it is being stored. So really, that carbon cycle on Earth is sort of one big carbon recycling system, and the rate at which we're using the fossil fuels and working with our climate essentially means that carbon's out of balance, and there's more carbon going into the atmosphere than there was in the past, and that's what's leading to sort of this climate change and global warming.
And Bex, I think you're going to share with us, is the same thing happening in the ocean? Or what are we seeing there?
Yeah, thanks, Jamie. So it's not dissimilar to what's happening on the land. So the ocean is taking up CO2 from the atmosphere, and we have phytoplankton and algae in those surface waters that take in that CO2 when they photosynthesize. They get eaten by bigger organisms and animals, and so that carbon moves up the food chain. But they also respire CO2 as well, so they're releasing the CO2 back into the ocean. When they die, their bodies sink to the sea floor, and they trap that CO2 into the sediments and lock it away. Not only do we have the animals in the ocean, we also have vegetation. So similar to what we have on land. They take in CO2 from the atmosphere if they're in the intertidal zones, or from the ocean itself. They lock that CO2 away in the sediments. And so that's a similar process to what happens on land, but in the oceans.
Brilliant. Thank you both very much. I think just to give a sort of a thought experiment for classes watching, I think we're going to see a slide here of the different carbon stores, so where carbon is around our planet. And this is going to be a quick test. This is just to see sort of how you think about this, but you can see along the bottom, you've got atmosphere, you've got fossil fuels, ocean, soil and sediment, and vegetation. And a poll has come up. If you got it full screen, don't worry, but just think about what order of size, what do you think is the biggest store? And the squares you can see are proportional to the sizes. What's the biggest store? What's next? What's yellow? What's blue, red, and grey? Think about connecting those names together. And the boxes, we'll give you 30 seconds to discuss in class, and then we'll come back and we'll reveal some of those answers. Brilliant. I wonder how you got on. I've got some great suggestions coming in. We've got a few different things. Vegetation coming up quite well on top, soil and sediments in the middle. But I think we've got a slide. I think, Richard, you're going to take us through this. Where is the carbon on this planet, and what's been happening to it?
Yeah, well, so oceans have our largest carbon store here on Earth, so large that they can't actually be sort of fully represented as a circle here on the slide. So, and you can see from pre-industrial times to current stocks, it hasn't changed massively. There's a bit more carbon sort of locked away in the oceans now. But coming up sort of in a close second, or not a close second, but yeah, the next one is soils and sediments. They have a really large carbon store too. So you can see how important oceans and sediments are for our global carbon stocks. You can see here that for the fossil fuels, represented by the black circle there, that's almost halved. That's gone down quite significantly since pre-industrial times to current carbon stores. And that's what we were talking about before, how we're essentially using them faster than they're being generated. And in line with that, you can see that atmospheric carbon stores have gone up quite significantly in those sort of past 200 or so years.
Brilliant. Thank you both very much. We're now going to hopefully that's given you a grounding in terms of carbon stores, the carbon cycle, but really important to have that, because we're now going to discuss the role of some of that ocean vegetation that Bex was mentioning in this next section, where we're going to learn a little bit more about seagrass, which is the focus for today's lesson. I know we've got some wonderful video of seagrasses around, and Richard, I think you were going to share with us sort of what is this habitat, what's going on, what would it look like if you were swimming in it?
Yeah, sure. So our role really as part of the Convex Seascape Survey is to understand how these seagrasses and other blue carbon habitats can help really in reducing atmospheric CO2. So here we have seagrasses. We have sort of a particular focus because they're such great carbon sequesters or storers. So they're storing that carbon that's up in the atmosphere, and they're doing this much, much faster than the rainforest. So that's great and a great sort of perspective for the future. And CO2 is being absorbed from the atmosphere into the seagrasses, and then it's being locked away in those sediments that we talked about before. There are also they exist sort of in intertidal zones, so areas of close to the shore. So when the tide's in, they'll be underwater, and then when the tide goes out, they'll be exposed. And they typically are around 4 meters away from the coastlines, so they're much easier to identify, much easier to sample from. But also that proximity of them to the shore means that they are susceptible to changes of land use and from human disturbance as well. So they're great habitats. They also provide this sort of wider biodiversity benefit as well. So not only have they got this great role in terms of carbon stores, but also they provide homes for sort of young fish and smaller marine animals as well. So not only should we be protecting them for carbon, but also we've got to think about their role in biodiversity as well.
Brilliant, Richard. Thank you very much. And am I understanding it correctly that they're drawing down this carbon from the atmosphere low tide when they're exposed, from ocean at high tide, but that's also going into the sediment. It's not just being stored in the sort of the tissue of the plant itself.
That's right. Yeah. So that carbon is sort of exchanged through the water into the seagrasses, and that carbon's going into the plants. And like you say, that carbon then is going into the sediment. But that's a lot of where our knowledge ends. And really what we want to understand is how far really does that carbon is exposed around the perimeter of that bed? Is it just within the perimeter of those seagrass beds? Or is it actually are we storing carbon through those seagrasses into much wider areas around the seagrasses? And this obviously has implications of how we promote and manage seagrasses in the future and how we should be managing them as well.
Bex, the seagrass is a type of what's called a blue carbon habitat. Seagrass is one of those ones we have here in the UK, but I think we can see a few others. Just briefly, if you can tell us sort of what they are and whereabouts, whether they're sort of UK, global, just tropical, would be fantastic.
Yeah, of course. So, as Jamie said, seagrasses aren't the only blue carbon habitats that we have around the world. So we also have salt marshes. So these are our coastal wetlands. We also have mangroves. So these are usually found in our tropical areas, and it's the only blue carbon habitat that we don't have around the UK. We also have kelp. So that lives in our deeper waters, and of course our seabed, which we've also been talking about.
Brilliant. Thanks so much. I mean, I find it amazing because we think a lot about conservation and nature conservation. If you look at a map of the UK, and suddenly when we talk about the continental shelf, you see, okay, wow, maybe the UK is sort of twice as big or 3 times as big as we think it is. And there's a poll for students to think about carbon stored in UK trees versus the UK seabed. And I would love to know, classes, you've got 30 seconds to discuss this, whether UK trees store twice as much carbon as the seabed, whether the trees and seabed store the same, or whether the seabed stores 3 times as much carbon as trees. So have a think, and we'll get back to you in about 30 seconds. Well, a lot of you are super scientists coming in with the seabed storing 3 times as much carbon as trees. It is quite amazing, and it really asks us to reflect on our conservation priorities in the UK and further afield. I mean, blue carbon it is quite amazing. You touched on, it's great, but just share with us some of the sort of top things that you think classes should know about these habitats.
Yeah, sure. So yeah, obviously within the UK and globally, there's a range of these different blue carbon habitats, all playing this pivotal role in increasing our seabed carbon stores and reducing atmospheric CO2. So the reason that blue carbon habitats play such a pivotal role is because they're capable of storing carbon much faster, so more than 10 times faster than rainforests. They're capable of storing carbon as much as 5 times as more carbon per unit area as our forests, and that carbon is effectively stored within in the sediments too. So our role really is to understand how much and where this carbon is stored to ensure that we provide the data to ensure that carbon is protected for generations to come.
Amazing, thanks so much. And it's a really great question because there's a finite amount of resource going into conservation. Want to protect everywhere, but we have to make choices. You've got a special method, and I think this is the sort of next section, is to come up with how do we answer these questions, and it's called eDNA. There's a student sheet. One side of it has some scenarios, which we'll talk about later, but the other side has the sort of 5 steps that Richard and Bex are going to talk through. But before we get into eDNA, can we just start with DNA? What is DNA?
Sure. So DNA is your what we call the building blocks of life. It's essentially the guidebook for how organisms function and how they exist. And we call this guidebook, it's called a genome. And it's comprised of just 4 letters, what we call A, T, C, and G. And it's that unique combination of these letters that make every organism unique. And what's sort of extraordinary is every cell in your body has your entire genome. And your genome is, for human genomes, is 3 billion letters long. So every cell in your body has this guidebook for how you function as a human. And all organisms have a version of DNA. The DNA is different from species to species. And you might be familiar with DNA for other investigations. So police investigations, for example, you've got sort of crime scene analysts come in and they use DNA to identify who's been at a crime scene. And that's exactly the same approach that we want to use here. We want to use DNA analysis on these seabed sediments to work out which organisms are leaving their carbon here in the sediment.
And that's eDNA, is it? We're not taking it directly from an organism. The E standing for exciting?
If only, yeah. So environmental DNA, or eDNA, it differs slightly to DNA analysis. Basically, we don't have the organism present. So if I wanted to sequence your genome, Jamie, I'd take a sample from you and I could sequence it. But for eDNA, we're analyzing cells that have shed from that organism, bits of plant debris, fish scales, those sorts of things. So what people might not realize is that you're constantly shedding these cells from your body, and you're leaving a trace of yourself in your environment. So as you walk through your classroom, through your house, you're shedding hair and skin and saliva, and all of those cells contain your DNA. And we can use these eDNA methods, highly sensitive methods, to sample that environment and pick up those traces to work out where you've moved within an environment and who's been there. So that's exactly the same as we're doing in these seabeds. We can pick up trace samples from animals, fish, but importantly for our questions, plants, and work out what's contributing to those sediments.
So essentially the question we're sort of thinking about, and I think we see them here, is that we know that we're probably going to find seagrass DNA where seagrass is.
Yep.
But if it's further away, we're going to be able to answer some other questions.
And that comes into sort of our main questions. You've got your seagrass beds, and like you say, we're expecting to find DNA from those seagrasses in the beds, but how far away from those beds do we find the DNA? And if we're finding it 50 meters away from the seagrass bed, then we know that carbon that's being stored or sort of generated from the seagrass beds is contributing to that wider environment, and that helps us understand where that carbon is and how we should protect it.
So I think you're going to take us through the five steps of sort of eDNA analysis now. Uh, Obviously, you've got to get out there first. So take us through it.
Yeah, of course. So step one is we go out and sample. So this is one really fun and exciting part of our job is that we go out on boats and we take sediment cores. So we push a tube into the sea floor and we pull up all these muds and sands that are from the seafloor. So we take a core or multiple cores from inside that seagrass bed, and then we also take cores from outside that seagrass bed as well. And then we take these cores back to our labs in Exeter. And we know that the samples have all the material that includes the DNA, but also shells. We've got muds, we've got sands, maybe little plant bits. And what we want to do is target that DNA. And so we put a sample into these little tubes and we need to break apart the cells which contain the DNA so we can target it and find out where it's coming from. So we have to physically shake it really fast and aggressively to open up these cells. And yeah, we can start exploring that DNA. So it becomes a bit like a soup of everything in there. And again, we want to just make sure that we only have the DNA in there. So we go through chemical processes to get rid of the stuff that we don't want. And we use really sensitive lab equipment to do that.
So first of all, you extract it, you're sort of bashing it around, breaking it out of the cell walls, and then there's this sort of purification. So there's probably, as the soup, you just want the particular bit of the soup.
That's right. Yeah.
Amazing. What's next? Now you've got your DNA. What do you do then?
So we then go through a process, what we call sequencing. So we talked previously about genomes and this being this sort of guidebook for how an organism functions. And as I say, that if you imagine that genome as a whole book, this process of extracting is essentially tearing pages out of that book and pulling out sections of that book. It's just part of that process. So our DNA is getting shorter. So it's not in these huge sort of 3 billion letters long now. It might be into a few hundred, a few thousand, but they're still in orders of those A, T, C's, and G's. So again, if we imagine that as a book, you might just have a paragraph out of a book. And the biggest challenge we have with these eDNA is that we don't have just one organism. We've got multiple different organisms. So a bit like we're talking about with the books, we've got multiple different books and we're ripping these all up at the same time and mixing them all together. And we're reading them section by section. But we're like with all technology, it's really increased and improved over the sort of the past decade or so. So the human genome that people might have heard about, that took over 20 years to complete, and it cost over $2 billion to sequence it the first time. Now we can do a human genome in a few hours, and it costs less than $1,000. So it gives you an idea of how quickly and how fast this technology has improved, and we can use that technology here as well. The other big change as well is that sequencers have changed in size. So they used to take up a full room. Now we have these handheld sequencers. They fit right in our pocket. So we can take these sequencers out into the field. We can take our DNA, like Bex has mentioned, and we can actually add it straight to the sequencer. And in the field, we can perform this analysis and get our data in the field. And that's really important because it means that we don't have to worry about transporting samples back. We don't have to worry about degradation of samples. We can do this live in the field, and it's all of these improvements. So you might be seeing images of sort of green squares flashing on a screen, and that is live pieces of DNA going through one of these sequencers. So when it's a green color, that means a molecule of DNA or a section of this book, if we stick with that, is going through our sequencer and it's being analyzed. And we get to do that in real time. So we can read our DNA in real time and we can get these answers and understand what organisms are there. But that's only really the middle, if you like. So we talk about all that lab work that Bex has talked us through, and we've talked about the sequencing. The other real challenge with eDNA work is this identification piece. So we've got our DNA sequences, that's great. We've determined the order of the letters, but how do we identify which organisms they've come from? So again, if we stick with this idea that our genome is a book and we've sequenced pieces of them, we need to essentially go to a library and identify where that page or where that paragraph has come from. And that's difficult. That's really computationally intensive. And all those advances we've seen for the DNA sequences, we've seen in terms of computers as well. So we use high performance computers and increasingly AI as well to help us understand and classify that DNA. And a bit like if you were taking a section of a book, some parts will be quite repetitive. It might have a lot of ands and the's and be very sort of general, and it's really difficult to identify. And you might go, okay, well that's in English and it's talking about here, and that's as far as I can get. I can sort of it's this section of the library. So we might be able to go, okay, that's an animal or that's a plant, but we can't tell any more information. But it might be that we sequence some parts of the DNA and it's so unique to that organism that we can really identify where it's come from. And that's what we're trying to do. And the deeper we sequence, and by that I mean the more sequence data we generate, the more data we acquire, it just helps us build up that picture of which organisms are sort of contributing to this. I guess the other thing to add very quickly is it takes time. So this classification can take hours, sometimes days. It's not quite as fast as sadly television makes it out to be. It's far less glamorous and far more intensive. But it's the same process that you see there.
Richard and Beck, thank you so much. Absolutely fascinating. Have a much better understanding of eDNA now. I think in this last section, and then we'll be able to move on to questions. We're just going to sort of put these sort of everything we've talked about today into a couple of sort of scenarios, conservation scenarios. And if we're thinking, you've told us that seagrass is amazing, but we're not quite sure where the carbon is stored from it, whether it's locally or more widely. So if a charity or a government agency wanted to plant more seagrass, how could we go about it? And there's a student sheet. You can see two sets of graphs. This is kind of sort of, and I know you've sort of made this simple for us to understand, which is incredibly kind. I know you do incredibly complex work, but we've got the sort of first scenario, and we're going to make our students to make some decisions about what should be done. We've got the first scenario on screen. Can you just explain to students what they're seeing there?
Yeah, of course. So our first scenario, you can see now. So we're looking at the DNA in the samples moving away from the seagrass bed. So we're starting immediately in the seagrasses and moving progressively away. And we're looking at the seagrass eDNA compared to all of the other eDNA we see in those samples. So immediately in the seagrass bed, so at 0 meters, it's basically entirely made of seagrass. And then we slowly get less and less seagrass in those samples as we move away from that bed.
Brilliant. And there's three choices that classes have to make. So whether we should just plant as much seagrass as possible everywhere, down to sort of the 4 meters you were saying, Richard. Conserve what we currently have because they've got this wider effect, or create conservation areas so we're guarding the sediment as well as not just the seagrass. You can choose more than one. So just discuss that, and we'll probably just do 2 rounds of 30 seconds because it's a little bit more complex than the previous polls that we have put to you. few people saying plant as much seagrass as possible, but sort of a 50-50 split for most people on conserving our current seagrass meadows and establishing these wider conservation areas to protect the sediment. Would do you think, is that what the data that you shared, would that suggest that?
Yeah, it would. So we're seeing quite a lot of that seagrass away from the immediate seagrass bed. So it would be a really good idea to start looking after and protecting those wider areas, not just the seabeds themselves.
Brilliant. So we're just going to see, we've got a second scenario, and we've got a graph which looks a bit different. So can you share with class of what's going on here, and then we'll go through a similar process for having a chat in classes for a minute.
So in our second scenario, we can see that in the seagrass beds, so this is at 0 meters, that all of the eDNA, or basically all of it, is from the seagrass itself. And then again, as we move away from the seagrass bed, that really drops off considerably. So the eDNA from the seagrass is just not there.
Brilliant. So we've got the same three choices again. We've got a minute counter coming up, and just to get a sense whether you'd stick with the same strategy or whether you'd change a bit. So we've got a slight different balance this time round, with a lot of you going for planting as much seagrass as possible and also conserving our current seagrass meadows. Does the data support that flip in choices?
Yeah, it sure does, Jamie. So it looks like it's really important to plant lots of seagrass, but it doesn't mean to say that we shouldn't be looking at conserving those areas around the seagrasses as well. So that's still important, but planting as much as we can is looking like a good option here.
Brilliant. And I know you're still in the middle of processing and sampling, so providing this type of data much more sort of sophisticated probably, but providing that data, and then that data can go on to inform wider decisions about how we look after our seabed and ocean. When might we hear what are the next steps?
Yeah, that's the that's the aim. Like you say, is that we want to provide really robust data to inform that policy for making those decisions out of those two scenarios, and the best way to do. So yeah, we have people in the lab right now, actually doing this type of work, performing eDNA on sediments that we've collected from not just around the UK waters, but internationally as well, really to answer these questions. So we're hoping well before the end of the Convex Seascape project to start informing policy and understanding the role of these seagrasses in our carbon stores.
Amazing. Thank you both very much. We've got some wonderful questions coming through from classes. Please do post more and more. You'll also see some of you have found the thumbs up button beside questions. I do tend to pick those ones which have been thumbed up more to go first. And do check as well to see if somebody else has asked the same question, and you can always thumbs up theirs, and we can make sure that gets answered soonest. So the first question is from Ned, who is home educated. Ned would like to know, is seagrass different from seaweed?
Maybe a question for you.
Okay. Yes. Yes. Is the answer. I believe seagrass is one of the UK's only sort of marine flowering plants. So it's a sort of a different taxa. But yes, it is different to answer to seaweed. But all of them playing an important role in sort of in the oceans.
So one's plant, one's algae. You can
Yes.
look that up a bit later, but different. Perfect. Next question. This is from Joseph, who would like to know: does coral store carbon too?
Yes, it does. It stores carbon, but not organic carbon. So it's just a different type of carbon. So the carbon in the ocean is made up of lots of different pools. So we have organic and inorganic, and the corals sort of produce this inorganic carbon. It still takes it from the ocean as it builds its shells, but it's just a slightly different form of carbon than what the seas have.
I think this is an important clarification when we're talking about blue carbon habitats that coral is an animal. We've been talking about plants and algae.
Exactly.
So just to remember that one. Amazing. Next question is from Alex. What would happen if we didn't store carbon?
That's a brilliant question. Essentially, if we just kept going in, it's sort of kept going into the atmosphere. Eventually, yeah, like we're seeing now, but just more pronounced. So temperatures would rise, and we would just sort of see overarching ecosystem collapse. But yeah, essentially, if we just kept raising CO2 levels, yeah, there'd be other things happening too, but that would be the most dramatic.
Brilliant. Thank you very much. This may still be a progress question, but Dravan would like to know which sea in the world stores the most carbon.
That's a question.
Yeah, that's $1 million question, isn't it? I guess our biggest ocean is probably the Pacific, so it has the largest capacity of storing carbon. But that doesn't mean to say that areas that have a lot of these blue carbon ecosystems don't also play a big role. So it's sort of balancing up how much carbon can be stored in the ocean and also along the coast as well. So yeah, it's still an outstanding question, but something we're trying to answer.
Brilliant. Great question there. Darian would love to know, how does carbon dioxide get trapped in salt marshes?
Salt marshes aren't an area I know much about, if I'm honest.
No, it's not something that either of us really work in. They're quite specialized ecosystems. So there are scientists who work in specific areas. So it's not something that we work in, but we can look into it.
I guess the CO2 isn't only coming in through the plants, and you'll have plants in salt marshes, but you'll also have all those microorganisms and other things appearing in that as well, who are playing an important role. So yeah, many different routes, if you like, for the CO2 to come into ecosystems.
Fantastic. Thank you both very much. This is from William. Is seagrass one of the most important ways to stop global warming?
I think they're all important. I think we have to. I think we can't sort of put all our eggs in one basket, for want of a better analogy. We have to really leverage all of these different things. We need to manage these ecosystems better. We need to protect them. We need to understand them. I think seagrasses, as we've identified, are really important. In the UK, especially, we've lost, I think, sort of 90% of our seagrass habitats, and it shows you really sort of how decimated they've been. But not everywhere has seagrasses. So it's, again, working locally, understanding what blue carbon habitats that we have, which ones can we use. But that comes from understanding. And once we have that understanding of how they operate, we can introduce policy to protect them and sort of, inform governments and others to help with that. So I'd say they're all equally as important. But in different areas, different things.
Brilliant. Thank you very much. This is from Fallinge Park High School in Rochdale. And they would like to know, how long does seagrass live for?
I don't think it's that long. It's a few years, from my understanding. But they release seeds and they can keep blooming in the same areas. So they have like quite a short lifespan in comparison to something like a mangrove. but they self-seed, so they can sort of like sustain their beds as well.
So I think if the habitat that they're in sort of sustains them, they'll continue to grow and expand. That's what we're looking for.
We've got some more great questions from that school. So we have how much oxygen does the seabed produce? And then the next question will be about, but let's go for that one first. Does the seabed produce oxygen, and if so, do we know how much?
There will be in terms of that sort of photosynthesis type thing. You'll be having oxygen produced. The exact numbers, to be honest, I don't know.
I don't know either.
Yeah, but yes is the short answer. Quite how much is something that we should look more into.
I'm going to come back to the second part in a bit, but we just had a couple of questions upvoted. So we have, how long did it take to complete your studies to become an expert in seagrass and eDNA? Obviously, some people are looking to follow in your footsteps here.
In terms of the eDNA, for me personally, it's been multiple years, and I think really it's keeping up with the innovation. So and it's improving so much, it's constantly improving. So, and it's becoming far more accessible. So, and that's really what attracts me to some of these studies now, is that I've always been sort of involved with environmental work, and it's now we can take these tools that we had in the lab out into the field and answer some of these questions together rather than trying to do them separately. So, I've been using these tools for sort of 10, 15 years. And they're constantly evolving. So the way we extract the DNA changes, the way we sequence the DNA changes, and the way we analyze it sort of computationally is also changing. But it's really exciting. And that doesn't mean that you have to do 15 years worth of work to get into it. Just in a few days, you can learn so much. It's far more accessible now.
I mean, you're both doctors of science. You've got your PhDs. That's at least 6, 7, 8 years of studying at university degrees.
Yep.
Yeah.
If that's quite a commitment, are there other roles, just to follow up from that, you can start to do this work without, saying, okay, you've got 8 years at university as a minimum before you get to your position?
Yeah, no, absolutely. And I think that's as I say, the real benefits and those technological advancements is you don't need to have big university labs with all the resources and infrastructure to do these things now. As the sequencer I showed earlier is accessible to anyone. Anyone can buy that, whether you've got PhDs like we do, or you're at a smaller company wanting to do biodiversity type research. The tools are out there. Obviously, there's so much of a community around this online as well that you can tap into and sort of understand, and the ability to share techniques and knowledge really drives this field forward. So it's really hopefully becoming more and more accessible to everyone.
And just we had a question about equipment, which I'm just going to sort of focus that in. How much would the science department need to sort of raise to get one of you? And do you need anything else?
So this one now they're holding here, this is it's all in dollars. So this one is $1,000 to buy, and it's as a starter pack. It's from a company called Oxford Nanopore, and that comes with everything you need to be able to do sequencing where you are. In comparison, so we're at the university, we have still some of these big sequencers, and they cost $1 million. So it gives you an idea if that's your sort of medical size grade equipment. These are now far more accessible. So hopefully, and as I said, I think that's all envisaged to try and get into schools and get into a sort of an earlier stage.
Brilliant, thank you. I'm lost track of time. We've only got a minute or so left, and we've got lots of wonderful questions. But this from Python Creek Primary School, wonderful. Is there carbon all over the seabed or only on the continental shelf? Because I mentioned, I think, in my introduction that part of the ocean.
Definitely a question for that.
Yeah, so it will accumulate on the continental shelves, but we have ocean currents, we have big like landslides, but in the ocean that will then transport all of that carbon offshore into our deep oceans. So this could be up to 3,000 meters of water depth. And that is a really good store for our carbon. So it really locks it away from the atmosphere.
Brilliant. And then there's a wonderful question, and which you may or may not, was great. I'm just going to ask it. What would happen, and it's a final question, very sorry, but what would happen if you found a new species when looking at the eDNA?
So that's the dream, because you get to name it. So That's it. So you find a new species, and this has happened. Not on sea, but on land. People have sequenced so far back in time that they're identifying species that don't exist anymore. So we've got so much sequence data about them that you can identify new species. But yeah, that's the thing. If you find a new species, or even with microbes that we haven't identified yet, the person who finds them gets to name them, and that's the fun bit of the job.
That's
We've
exciting.
got to come up with a name. So.
Richard and Bex, thank you so much. Absolutely fascinating. A whole new world for me, and I imagine a whole new world for the classes watching. The importance of the ocean, blue carbon habitats, the research that you're doing, eDNA, conservation priorities, science and policy. Absolutely brilliant. So thank you very much for all your insights and enthusiasm. Thank you very much for all those classes who have taken part as well. Hope you've enjoyed it as much as I have. Until the next time, it's goodbye from us. Bye-bye.
Thank you. Bye.
Bye. Thank you.